My name is Amy Klippinger, and I direct the PETA International Science Consortium. I'm joined today by two co-hosts, Dr. Dave Allen, who is President of Integrated Laboratory Systems and the Principal Investigator for the NICEDM Support Contract, and Dr. Stephen Moorfield, who is Senior Project Manager at Integrated Laboratory Systems and the Project Manager of the NICEDM Contract.
The goal of today's session is to facilitate the use and regulatory acceptance of non-animal approaches to assess inhalation toxicity by identifying factors key to the design and use of these approaches, as well as remaining gaps. As you're listening to the speakers today, please keep this in mind. We'd like to walk away from this meeting with a better understanding of when and how non-animal approaches can be used now and concrete next steps to expand their applicability. Okay. As you all know, there are many initiatives underway to develop human-relevant approaches to predict the potential health effects following inhalation. And we certainly don't have time to talk about all of these today, but since we did not have SOT, I wanted to highlight the e-posters that are online. If you go to the SOT app and click on e-posters here, there's a lot of good information in there, and I've just put two here that I think would be relevant to this group, a poster by 3M and one by Cytotoxicity on in vitro testing at the air-liquid interface of different chemicals. So if you haven't had a chance, it's worthwhile to check out these e-posters. There's some really good stuff on there. To quickly review the order of events, you can all see this on the agenda that was emailed around yesterday. We'll hear from three speakers, and then we'll take a couple clarifying questions after each of these three presentations, but we'll save most of the questions and the discussion for the hour that we have at the end. We also emailed around a little cheat sheet yesterday from each of the speakers, which was meant to facilitate today's discussion. So this will hopefully avoid some of the clarifying questions about the experimental details and so forth, and just save us a little bit of time. We will then have a 10-minute break after we hear from our three speakers, and then we'll hear from the U.S. Department of Defense, the EPA Office of Pollution Prevention and Toxics, and the Office of Pesticide Programs, who will discuss these approaches in a regulatory context and help frame the discussion. And then that's when we'll have our about an hour discussion at the end. You all also have the discussion questions. These are at the end of the agenda that was circulated yesterday. We expect to have about 40 people on the line today, so we're going to do our best to have a discussion. There are two different ways that you can ask a question. The first is to submit it using the chat box, which should be in your WebEx toolbar. And you can send those in at any time, and either Dave or Stephen will read those out loud. Or the second way is you can use the raise your hand feature if you actually want to say your question or comment out loud. You should see a small hand icon to the right of your name in the toolbar, and that's what you can use if you'd like to raise your hand. So please use one of these two methods, and like I said, either Dave or Stephen will read your question or call on you. So we're going to try to prevent everybody from talking at the same time. One other practical matter I mentioned already, please try to keep yourselves muted if you're not talking, otherwise we get a lot of feedback on the line, especially with this many people joining us today. And lastly, we will be recording the presentation part of this session today. We will turn that off for the discussion at the end, but we'll have these presentations recorded and we'll post them online if anyone's interested in listening to them later. So I will now introduce our first speaker, but before I do that, I want to check in with Dave and Stephen to see if there's anything I missed or anything either of you have to add.
Thanks, Amy. Just a couple quick administrative things. So Stephen, as you may have noticed, has muted everyone other than the presenters. and there is a mute microphone icon beside your name. You can unmute yourself using that or by dialing *6 on your phone, but that's to control background noise, obviously. Also, as Amy mentioned, if you have questions during the presentations or during the discussion session, please use those features she mentioned. If you use the chat window, I would ask that you either send the question to everyone or to Stephen Moorfield directly. If you send it to anyone else directly, we won't see it.
Okay, thanks, Dave. Then I will go ahead and introduce our first speaker. First up is Dr. Adam Speen, who is an ORISE postdoctoral fellow working with the EPA Inhalation Toxicology and Facilities branch under the mentorship of Dr. Mark Higuchi. Adam received his PhD in toxicology from the University of North Carolina at Chapel Hill in 2018, researching drug-environment interactions and ozone-induced health effects on human lung tissue.
Okay, good morning, everyone. Let me share my screen here. Okay, so thanks very much to Amy, David, and Stephen for organizing, and obviously they can let me know if at any point I cut out or you can't hear me or you can't see my slides or anything. But it's really great to be in the proverbial here, I guess, today. So I want to say that I'm presenting a lot of work from a lot of different people, particularly from the group of Mark Higuchi in the Inhalation Toxicology Facilities branch at the US EPA. And let me just get out a pointer for you all here. All right, great. So first of all, a disclaimer, right? The research here has been reviewed by the Center for Public Health and Environmental Assessment at the EPA, but none of that signifies it reflects the views of the agency or any mention of trade names or commercial products don't produce endorsement or recommendation for use here. So first of all, I want to talk about the main objective of what our study is doing. So one thing that's probably not foreign to many of you is the utilization of direct exposure on cells at ALI. That's what a lot of us are trying to do. I think some of the novel approach of this research is actually the generation of this transcriptomic data set in order to identify concentration-dependent mechanism and mode of action in airway cells. To break that apart a little bit more, I think that a big part of it, and some previous work was done by my predecessor, Jose Zavala, on this work, where it was a screening of volatile chemicals in this concentration-dependent method, actually creating the exposure system, validating it, making sure that it's working as we think it is, and a lot of this data is birthed from that hard work that he did. We wanted to assess the technical and biological reproducibility of this transcriptomic data for these air-liquid interface cells. Again, that concentration-dependent changes, I think, is a really important way of looking at this data, as opposed to endpoint cytotoxicity or viability endpoints. To be able to evaluate that transcriptomic data, to actually do some structural activity analysis. And again, this is something that I don't show today, but it's an end goal of being able to pipeline this transcriptomic data the same way that a lot of the high throughput submerged studies would do. And finally, be able to evaluate both the ACGI HTLV values as well as the concentration range for a lot of in vivo studies that are done. So is it even possible to compare some of this in vitro work to existing in vivo studies and how they're conducted? This slide is a brief overview, very similar to that info sheet that Amy put together, which I think is a little more thorough than just this one slide. So you can reference that later if you're looking for details. But just to summarize, we're using two cell types in the data I'm showing you today, primary human bronchial epithelial cells from volunteer donors, as well as a cell line, Bs2B cells. We actually screened 8 different chemicals in this study, 4 of which were done in both cell types. So those are the ones with the asterisks you can see over here. And you'll notice the data that was done in one cell type versus both cell types for comparison. We're able to do 6 concentrations and all sham control and an incubator control. And these concentrations are during the same exposure, right? So we're generating the chemical. We're actually doing a serial dilution, which I'll talk about in a moment. And they're done during that same exposure time window. The window we're talking about right now for this study was 2 hours, and most of our assays conducted 4 hours post. We do the transcriptomics analysis as well as measurements of cell viability and cytotoxicity. And all of these were done with an N of 3. So 3 different days of exposures. And in the case of the primary cells, 3 separate donors, actually. So let's look at the actual exposure overview. Some of you have probably seen this before, but I think it's worth talking about before we actually look at the data, just so you know exactly how we're doing our exposures. This is developed by Mark and Jose in the Inhalation Toxicology Facilities branch at the EPA. And if we're just looking at the right-hand side of the screen there, essentially what you have is a large box, almost a large incubated box where you're able to actually expose your cells as your sham control at the same time. So just to give you the overall workflow of how we're treating the cells before they go in, all the cells are grown at ALI. Obviously, they go ALI at different times depending on the cell type. The apical side is washed prior to exposure in order to make sure there's no mucus or anything that's going to inhibit the volatile chemicals from actually reaching the cells. We're adding Keepy's buffered media to the basolateral side in order to maintain the pH of the media in a low CO2 environment. From there, we actually take the cells to the exposure lab. They're placed inside one of these 2 chambers. So you have your VOC exposure chamber, and the plate looks like what it does right below. So you have A, B, C, D, E, F concentrations each. So 4 wells basically at each concentration. You have your sham control plate over here, and then an incubator plate, which is kept separate outside of this exposure system. We're just keeping that in the traditional incubator. Some nuance to the system, right, is as those VOCs are generated, they're entering a dilution tree and being mixed with humidified dilution air in order to actually achieve a serial dilution as that chemical moves down that dilution tree. So in this case, we're using half-log dilution, adding dilution air to the VOC source each time before it's actually sent to its respective section of the plate. And in the case of our sham, it's just receiving that humidified air. And one thing to note, and a thing that we've learned over and over again, I think, is that it's really important to keep these cells, try to keep these cells at a physiological condition, right? So this box is staying at 37 degrees C. It's hard to do that for the entire box, so you really have to monitor it very carefully. As well as I mentioned, that humidified dilution air, right? We're trying to stay above a minimum is around 75% humidity at any one of these concentrations going in. And so we're trying to maintain that throughout to keep it consistent for each of our exposures. The Yeah, you can see over here on the right, here's your cells at ALI. Here's actually the nozzle that enters into each one of those wells. So each 4 of those wells in a concentration has one of these inlet nozzles going in. Your chemical is moving through. It's actually pushed onto the cells, and then it's actually vacuumed out. So the cells are under a constant concentration the whole time, right? They're receiving constant concentration the whole time. And it is being monitored in real time. So at each one of these dilution T's, at one set is being sent to the cells. There's actually another inlet on the other side that you can measure the actual amount of chemical by GC MS. Finally, post-exposure, the VOC is exposures for 2 hours, and the cells are removed, and then samples collected at 4 hours post. I won't spend too much time talking about our in vitro models, since many of you are probably fairly familiar with them. Suffice it to say, the Bs2Bs are easy to grow, easy to replicate, and so we're at least able to get some consistent, repeatable data with them. And then the primary cells, again, I just wanted to highlight that these are from 3 different donors on each one of the days that we're doing exposure. So we can expect at least some variability there in these cells, especially because they're from different donors, as well as because they're fully differentiated with cilia and mucus-producing cells in there. So those cells have to be treated very, very carefully prior to make sure that the apical side is washed, healthy, and exposed at ALI. I don't want to walk through all of these VOCs that we're testing today, but I do want to just highlight that, right, all of these have been regulated or are regulated in some form or another due to risk of human exposure. And most of those, the reasons are those red highlighted terms in each one of the descriptions. And there's a lot of data out there pertaining to these when it relates to in vivo inhalational toxicology studies, LC50s, epidemiological studies, but there's really not a lot of info in terms of how it relates to air-liquid interface in vitro exposure. And that's really the crux of what we're trying to do. We're trying to see if our methods and our data really aligns with some of the existing in vivo and epidemiological data out there for these chemicals. I already mentioned before, but those analytical endpoints, right? We have an LDH cytotoxicity, the cell titer glow viability assay. So if you're not familiar, right, that's a measurement of just ATP generation in the cells. Both of these are really what we're using as our jumping off point. So we're using the cytotoxicity and viability data to kind of range find in terms of where we want to be when we do this transcriptomics biospider tempo-seq RNA sequencing data acquisition. And so as we'll look at these earlier, really we're just making sure that we're trying to stay below a 30% cytotoxicity or change in viability at the highest dose. I'll talk a lot about the Biospider tempo-seq platform and how we're doing our analysis. And then finally, I just wanted to mention, right, that as I said, we're sampling the chemical generation at usually 2 to 3 times during the exposure. So we have kind of a real-time data point of what concentration the cells are actually seeing throughout those 2 hours, which I think is a big strength in comparison to a submerged dose study as opposed to this concentration here. So the viability and cytotoxicity data, as I said, are really our jumping off point. So there's not a lot of exciting things to see on this slide as well as the next one. But I think that the main point is that I want to reinforce that the concentrations that we're using are below below overt cytotoxicity or changes in cell viability in these cells. So as you can see in all of these, and this is LDH release from the cells, it's only at that highest concentration and only in the Bs2B cells in the red that we even see any change in compared to the control. The only outlier might be that dichloromethane, which that high concentration, we do see some change in cell viability up there. The same thing for the cell titer glow data, right? So again, we're seeing less ATP means less viability in the cells. And again, right, there's a pretty significant change in the Bs2Bs at the highest dose for most of these chemicals, right? If you're looking at 1,4-butadiene at the top there, or 1-bromopropane, and almost all of the ones on the bottom here. Use that Bs2Bs around below a 30% change as our starting concentration. So the fact that we don't see it in the primary cells, it might just be due to them being more robust. You know, that's not surprising. That they might not react as much to being exposed to these different chemicals. But you can appreciate that we're at a pretty low change in cell viability, even at our highest dose. In most of our doses, we don't see really any change from the control. I'll try to move through our BMD analysis a little bit quickly, but I want to make clear for people who aren't using this type of analysis. So we're collecting a whole cell lysate. We're sending them to a third party, to Biospider, who are doing the TempoSeq assay. And they're sending us back basically the gene counts. So each of the targets that they're measuring, and we're seeing whether each target with a specific chemical at a specific concentration, is there a dose response there? And so just working from top to bottom here, right? Pre-filter, making sure that there is statistical significance and that there is a fold change magnitude that's actually large enough to be real or to actually be important. We're modeling. It's a continuous model chosen for the best fit. And so it's running through all of these different dose response models. The benchmark response factor relative to the control, and then the best model selection is done through that Akake's information criteria, basically, which is a statistical criteria to decide which is the best nested model to the concentration. And then finally, if it is a Hill model that is the best model here, it then does one more check, basically, to say whether that K parameter is in fact less than a fraction of the lowest possible dose, right? The lowest positive dose in this case, and so we discard those flagged models if they come up. So this is basically what we see for each one of the targets. So each one of these figures down here is one target. So this is a CYP24A1. These are cells, Bs2Bs exposed to dichloromethane. And this is just the dose response for that target at this concentration in those cells. And so each target is modeled to a different dose response curve. And as long as they meet those criteria, we then move into pathway analysis or gene set collection analysis. So briefly, we use that 3-5 rule. It has to contain greater than 3 concentration responsive genes within that gene set or that pathway, if that's the way you want to call it. The BMD is within acceptable range, but the concentration responsive genes have to account for 5% of the genes in that set. So we don't want to be looking at gene sets that only collect one target out of their entire library. And at the bottom here, you're seeing different gene set collections that people are analyzing off of. Right now, we're looking at, I'll show you data from the molecular signatures database, MSigDB-C2, which is kind of an agglomeration for all of these. Reactome is a specific one, and then Hallmark, which summarizes a lot of well-defined biological states or processes. But the main point is really looking at benchmark dose or benchmark concentration. So at the probe level, basically each one of these plots, each panel is a different chemical exposure. Each point is a concentration responsive probe, right? And then at the bottom is concentration or potency. And then the y-axis over here is looking at kind of maximum fold change from the sham control, which is that 0 in the middle. And so what you can really appreciate is that it's very chemical specific what sort of response you're getting, even across all of the doses. So a thing to appreciate, for example, on these ones, looking at acetaldehyde and formaldehyde, right? A lot of these points are left shifted, and the magnitude of change control is very low, and so there's not a lot of targets that are specifically changed when we're looking at benchmark concentration modeling parameters for this condition, especially in formaldehyde, as you'll see. I'll move along a little quickly here, but looking at the top, you have your gene set collection on the left-hand side, along with each one of those first 4 chemicals we're looking at. These were just tested with the Bs2B cells, tested in the primary cells. But each one of these values is essentially the BMC of the most sensitive gene set. So we took a gene set within this collection. We said, what is the point of departure or the dose where you're seeing that change happen? And as these are getting more stringent, right, the Hallmark is probably the most stringent gene set collection, right? That might have a little bit higher of a benchmark concentration. But the main comparison for today that I want to talk about and think about is the comparison to that TLV value, as well as at the bottom here, you can see just some representative in vivo studies that have been done previously. And I'll show a summary slide of this toward the end, really looking at the low L and the no L concentrations used in some of these studies. We see a similar thing for the other 4 chemicals that we tested. The main thing to look at here is the comparison between the Bs2Bs and the HBCs, right? Even in the 1-bromopropane, you can see far less targets meet our parameters and our criteria in the HBECs compared to the Bs2Bs when we're using those same concentration, so the same concentrations throughout the exposure. I think that this could be rectified, obviously, if we extend our concentration a little bit higher. We use the Bs2Bs response kind of as our range finding, and we use the same concentration for the HBECs later. And the HBECs being a little bit more robust, less responsive, it's not a surprise that less genes are popping up. But in some cases, in the dichloromethane, as we saw earlier in the cell viability data, there was a response at that high dose, and these profiles look pretty similar in both cases. Looking at the BMC, benchmark concentration of the most sensitive gene set, again here, 2 things to, I think, focus on, right, is that we're just looking at 1-bromopropane in the widest ranging gene set collection. The BMC in the primary cells is always, I think in most cases, if not always, a little bit higher than the Bs2B cells, which is not surprising since there's so many less targets that we saw impacted anyway. And we're also looking at that TLV value on the left, and to see that it's really fairly close or within an order of magnitude of that benchmark concentration of the most sensitive gene set for each of these chemicals. And we see the same for the dichloromethane, as well as the trichloroethylene here, and the carbon tetrachloride. So we're really heartened to see such a consistent response and almost an alignment between our benchmark dose, point of departure concentration, as well as a TLV value that we started with. So we feel really good and justified with using this as kind of our jumping off point to do this screening. Let's look at a full summary so we can actually look at some of these in vivo data to compare. This table represents all 4 chemicals that I'm talking about today that I showed you here. On the left-hand side, obviously, is the chemicals, and they're ordered by that ACGIH-TL. This is in ppm. And then really what we're looking at is almost the same rank order all the way across this table. So acrolein, which has the lowest TLV recommended level of exposure, the response in the Bs to B cells within an order of magnitude at our most sensitive, our BMC for our most sensitive gene set, as well as the low L for one of the more representative in vivo studies that we at 0.4. Same for 1-bromopropane, right? Fairly close, and the rank order really appears to be the same. So these are organized by TLV. You can notice that the BMC increases all the way down, with one exception being that trichloroethylene right here, but still fairly close to our, actually, sorry, it's the acetaldehyde, which is our outlier in this case. But still within an order of magnitude from the TLV to our BMC. In the HBECs, we had less that we measured, and one that didn't have any BMCs that met our criteria. But again, the rank order is the same. And even in the low L and the no L, you can see almost an increasing concentration going up as we're looking down this list here. So I think that this is a really interesting way of approaching this data, since the way we designed this experiment was meant to mimic what Tox21 and what ToxCast wants to do in order to pipeline their toxicity and transcriptomics data. But the question is always whether it pertains at all to what we're seeing for human health, as well as these animal studies that are being done. And obviously, this is just a starting point, but we think it's really a really good one, and a nice representative study as a screen, especially to show that it might be possible to do some direct comparisons to these databases, which were built mostly on animal data. But if we're moving towards more in vitro methods, we want to be able to know if we can actually make those comparisons. Only a couple of moments left here, right? The lessons learned, higher concentration might be better for the HBECs. Obviously, the more immediate endpoints we see, like those cell viability and cytotoxicity data, helps inform how we're doing this transcriptomics analysis, since it's much far down the line. Humidity and temperature are very important, and then the timing for transcriptomic changes is obviously important. So I think I've talked about most of our goals throughout this talk. Obviously, we want to be able to use this as a better predictive tool. We want to be able to actually figure out how to do those comparisons between in vitro and in vivo data. And one thing I didn't mention, which I'll talk about right here, is that we want to be able to know how this works in our repeated exposures setting to be able to better mimic some of those animal studies that are done. Dosimetry, I think what's actually getting into cells is something we need to look into more. And finally, I'll leave you with this slide where this is kind of our next steps. We're taking out the Bs2Bs, adding 16 HBEs. We're also adding those MatTek epi airway cells as another comparison point here. We're adding some different aerosol exposures to see what sort of response they have. And a lot of the chemicals in this group that we're testing right now are really meant to compare portal versus systemic toxicants and how they actually change our dose response. Finally, we're adding that tier as well as some inflammatory response markers to this next stage of the study. So I'm definitely out of time, but I want to thank everybody for your attention this morning. I hope everybody's doing well, and I just want to thank all the people at EPA, ORD at Sophia, and CCTE that's doing this work. It's been great. So thank you very much.
Thanks a lot, Adam. We have time for one, maybe two clarifying questions. And of course, we'll have time in the discussion session.
Dave, I've gotten one question in via the chat. How isolated are each of the wells for exposure? Is there any chance of spillover from the wells, especially at the higher concentrations?
Yeah, it's a good question. So each one of those sets of 4 wells within a concentration, they're basically getting one concentration, which is split into 4 separate nozzles. But each one of those wells has its own. So basically, the inlet nozzle is sending the chemical in, as well as directly outside of it is then vacuuming it out of each one of those wells. So we're fairly confident that each one of the wells is getting the same concentration. Between sets, so from like concentration A to concentration B, the chamber that they're in is designed such that it should be very sealed around. It should be very tightly sealed around each one of those concentrations. So we haven't seen any spillover. We would see it in our GCMS measurements, and we're making sure that the dilution curve makes sense based on how much dilution air we're adding in. So we haven't seen it at all in the volatiles. The aerosols are maybe a little bit different story, just in terms of how they're acting in the exposure system. But for the volatiles, it seems to be pretty consistent.
Okay, Amy, I'm not seeing any hands raised or any other chats. So if you want to go on to Sandra.
All right, thank you very much.
Okay. Thank you, Adam. Next up, we have Dr. Sandra Vesterlen. Sandra studied biomedical sciences at the University of Antwerp in Belgium and then obtained her PhD in 2010 from the University of Ghent. She is an air-liquid interface project leader at Vitro, coordinating strategic and contract research in this field, and she's an she's an expert in vitro assay development and validation using human cellular models and molecular technologies.
Okay, do you hear me well?
Sounds
We
great.
can hear you.
Okay, that's perfect. Thank you, Amy, for the nice introduction and to set up, of course, this online session. At Vitro, we can offer expertise in animal-free methods for inhalation testing, and today we will present 2 case studies of air-liquid interface vapor exposure. So just one slide of Vitro. It's the Flemish Institute for Technological Research with its headquarters in Mol in Belgium. And it's a strategic research center of Flanders, which is 1/3 funded by the Flemish government. And our activities on inhalation testing are part of the health department within Vitro. And on the right, some figures. So for example, in 2019, we had some 959 employees, and it covers 45 nationalities. And on the picture, you can see one of the Vitro buildings. And in this building, we do our air-liquid interface in vitro inhalation studies. So chemicals must be assessed for regulatory registration, and it often relies on animal testing using the OECD test guidelines. Of course, there is an increasing demand to implement in vitro alternatives in different research areas. And inhalation, you all know, it's one of the major routes of exposure to foreign materials, and it may serve as the target tissue and also the port of entry into the systemic circulation. So alternative approaches are needed to better predict human responses to inhaled substances. And this includes, of course, you need a relevant respiratory cell model, realistic inhalation exposure systems, and proper dosimetry techniques. On the left-hand side, you can see a picture of the air-liquid interface exposure. And this might be a promising method as an alternative approach. You don't need animals, it's less costly, less time consuming, it's more reliably mimics the human exposure compared to submerged or rodent exposure, and you can also do some mechanistic effects. So to do these exposures, you need, of course, some infrastructure, some exposure systems. And at Vitro, we have an air-liquid interface platform with 4 air-liquid interface exposure systems. We have 2 commercial Vitro cell systems with perpendicular direct flow. And on the picture, it's the number 1 and the number 2. We have one commercial system with mist generation. It's well known as the VitroCell Cloud. It's in the number 3. And we also have one in-house co-developed system with a laminar continue flow, and it's called the Navetta, and it's in the number 4. And today, the case studies we present, we used the direct flow systems of VitroCell, the 6.4 in the number 1, and the 2448 in the number 2. So the first case study focuses on testing Saline compounds, and this project is called INSPIRE and is sponsored by PETA. And the general goal of the INSPIRE project is to demonstrate the utility of an in vitro system to predict the likelihood of a chemical to cause effects on the respiratory tract, and also to rank chemical toxicity. And of course, this approach should be acceptable for regulatory and non-regulatory purposes. Generally, Saline compounds are not stable and are highly reactive with water. So for the first case study, we selected 3-ethoxysilane as the most stable one. This is an industrial chemical classified as category 2 inhalation toxicant based on rat acute inhalation toxicity data. And in this study, we used the B2B cell line, which was also presented by Adam. And it was exposed to various concentrations of 3-ethoxysilane vapor at the air-liquid interface using the VitroCell 6.4 module on the picture on the slide. So 3-ethoxysilane, we never generated it before. So we did first some initial tryouts to check the suitability of this compound to be generated in dry clean air as carrier gas by the capillary dosage technique. And this is a method that was actually a homemade method. A dry, short exposure of the cell was needed because of the reactivity of the compound with water. And the stability of 3-ethoxysilane vapor was monitored online. And on the next slide, you can see a picture on the right, which shows the experimental design of the 3-ethoxysilane vapor generation, and it's indicated by numbers. The number 1, it's a recipient. This was filled with the 3-ethoxysilane liquid, and it was placed on a balance. And then in the number 2, pressure was set onto the recipient by use of a second capillary. Then you can follow it until number 3. The liquid was emerging at the outlet, and it was retained by a cotton plug. Over there in the number 4, you have a local heating element. So 3-ethoxysilane was evaporated. And then in the number 5, a dry airflow transferred then the 3-ethoxysilane vapor to the glass distribution line, which was connected to the VitroCell 6-4 system. And then in the number 6, we could monitor the weight loss. So actually, different concentrations could be obtained by altering the weight loss and the airflow concentrations, and also by a total hydrocarbon analyzer for the lower concentrations. So in the next 3 slides, there are some technical details. So the 3-ethoxysilane vapor generation, it was successful, and thereafter we started with the biological experiments. In these 3 slides, I just pick out some small things. Amy sent around the tables with all the details, so you can check later. So as mentioned, we worked with the bees-to-bees cells. They were exposed to the air-liquid interface in the 6-4 module. It was a dry exposure, and it was, of course, at 37 degrees. We always have as conditions an incubator control. We expose to a clean air as control. We have a positive control, and we always take nitrogen dioxide for us assays. And for the 3-ethoxysilane vapor, we set 3 concentrations, 0.72, 25, and 85 ppm. This was set after a range finding study and also based on LC50 data. So we did a back calculation to the in vitro concentrations. We only did a short exposure of 1 hour because we had to do a dry exposure because of the reactivity. We had a post-exposure, a submerged post-exposure in this case, and also for the biological endpoints, we looked for cell viability, cytotoxicity, and we also checked for inflammation. We used a 10 Vplex from the Mesoscale discovery platform we have. And this is the last technical detail slide. For the chemical analysis, we did ICP-AES. It's a 6-4 module of VitroCell, so we have 4 replicates in each run. We did 3 independent biological experiments in total. And for the chemical, we had some existing in vivo data, and also some key events were already reported. So in the coming slides, we present some results. This slide gives an overview of the measured delivered concentrations. On the top, you can see the table of the concentrations of the 3-ethoxysilane in the cells. And beneath, you can see the concentrations in the medium beneath the cells. For the lowest concentrations, the 0.72 ppm, this was below detection limit, so we could not measure. And then for the middle and the highest concentration, so for the 25, we obtained in the cells 3.2 microgram. And for the highest concentrations, we obtained 17.9 microgram. And in the medium beneath, it was a bit lower. It was 2.5 and 6.0. And there, for the concentrations in the cells, we calculated a delivery efficiency of 14%. Then next, we checked for cell viability with a Press2Blue assay. The different colors in the plot represented 3 different biological experiments, and then you see for each color 3 points. So these are the 3 replicates for each run. On the y-axis, you can see the percentage cell viability, and on the x-axis, you can see the conditions. Nitrogen dioxide was our positive control, so you can see a reduced cell viability as expected. And then for the concentrations of 3-ethoxysilane vapor, you can see that there was a concentration-dependent decrease compared to incubator control and clean air. And this was statistically significant for the middle and the higher concentration of 3-ethoxysilane vapor. We did the same for cytotoxicity with LDH. Here you can see the increase in cytotoxicity, which was also statistically significant. We also checked for inflammation, as was mentioned, and we checked it for the two lowest concentrations, the 0.72 and the 25 ppm. And you can see for the 25 that there was an increased release of IL-6 compared to clean air and compared to incubator controls. And the same trend was found for other cytokines. You can see it on the right of the figure. For IL-2, for example, IL-8, IL-13. So these were the data on the inflammation. So globally, the data of the triethoxysilane vapor obtained in 3 different individual experimental runs indicated that the air-liquid interface exposure method is robust and reproducible. So additional work is now underway to test other silanes and to determine if this in vitro system can detect the decrease in toxicity that correlates with increasing carbon chain length, and to determine also the advantage of a 2D compared to a 3D. So in the next phase, we also will do some tryouts with 3D models. And also, it's not on the slide, but also to try out repeated dose exposures. And these results will be published in a peer review journal later. So this was the first case study on triethoxysilane. The next one, it focuses on testing petroleum substances and its constituents. We called it the Petrali project, and it's sponsored by ExxonMobil. Petroleum substances are also subject to regulatory registration requirements. They are volatile to semi-volatile. They have a low aqueous solubility, and they contain many constituents in low concentrations and with a range of different physicochemical properties. So actually, this makes in vitro testing for inhalation toxicity a bit difficult. So the ultimate goal is actually to develop an alternative method to replace the in vivo animal test for the prediction of human in vivo inhalation toxicity. And one of the steps, of course, in accomplishing this goal is first the development and validation of a generation facility to obtain the vapors from petroleum substances, and also the optimization and validation of the VitroCell 2448 exposure system, a picture on the right. We started with the optimization and the validation using ethyl benzene. So on the left, you can see a picture for the vapor generation facility. It's a bit adapted from the in-house developed capillary dosage method, which was presented for the silane compounds a few slides ago. So here again, you can see some numbers. Again, there is a vessel on the balance, and in the vessel, you can find the ethyl benzene. Then nitrogen in the two was set onto the recipient, and then the outgoing liquid, it was emerging at the outlet, and it was precisely dosed in a tank. It's a number 3, you can see. And this tank, it had a heated base plate, and so ethyl benzene dropped onto the bottom plate of the tank, and so it was vaporized and then mixed with humidified airflow. It was also mentioned by Adam, relative humidity is important to keep the cells alive. So we have here a relative humidity of more than 80%. The humidified flow was guided to a double-walled glass tube in the number 4, which was situated in a water bath. So again, temperature is also critical to keep respiratory cells alive. And then the exhaust was split. One side was to build up enough pressure, and the other one was connected to a heated sampling line in the number 5. And this was connected then to, on the other side, the picture, the VitroCell 2448 system. In that system, you can do a dose response. You have a simultaneous exposure to a negative control, the positive control, and 6 concentrations of your compounds you want to study. The generated concentrations, they were determined by combining microbalance consumption and used airflows, and also with an FID analyzer on the other hand. So I mentioned it. We started with ethyl benzene. It's a monoaromatic hydrocarbon used in the petrochemical industry. It's classified as maybe fatal if swallowed and enters the airway, and harmful if inhaled. The hazard information is derived from in vivo experiments, and it can also be used to calculate the exposure levels at which no health risks are expected. This is known as the derived no effect level, and it would be of interest, of course, to extrapolate the in vitro data to the in vivo situation and to check if it can support traditional risk assessment. So for this case study, the technical details are not on the slides and are also not distributed because we have written it down now in a paper, and it will be submitted really soon to a journal. So in global, we used not B2B but A549 for this study. We did a 4-hour exposure in the module, and we exposed them to about 30,000, 40,000, and 50,000 milligram per cube. The in vitro concentrations were based on in vivo LC50 data. And here, the first slide, this is the first result. We determined the doses which come into contact with the cells. We determined them by headspace GCMS analysis in stainless steel inserts, which were placed in the Vitro cell module. So in the table, you can see on the left the measured exposure concentrations, the generated concentrations. Yeah, it's about 50,000, 40,000, and 30,000 milligram per cube. And then on the right, you can see the microgram we can find in the inserts, the 24-well inserts of the stainless steel inserts. And we calculated thereof a delivery efficiency of 0.1%. This is quite low. This is dependent on the deposition efficiency of the system. The 2448 has a low deposition efficiency, but also, of course, the physical chemical properties of these compounds and the absorption of the compounds. And that's the reason why we had to generate high concentration to obtain some micrograms onto the cells. So now in the next slide, we will see if we can see some effects onto the cells when we generate these concentrations. So the first figure here, it's cell viability. We did this with a MTT assay. And on the y-axis, you can see the change in viability compared to clean air. And on the x-axis, you can see our conditions. So for ethyl benzene, you see the 3 generated concentrations. We presented the data with box whisker plots, and they visualize the range of individual data points per condition. The mean is indicated here as a blue square in the middle of the boxes, and you can see different colors, and they represent the different biological runs. And for these experiments, we had 5 independent biological runs, and for every run, also 3 replicates. So you can see 3 different colors. 3 different bullets per color. For the data, you can see that there is a decrease of cell viability from the lower to the higher concentration of ethyl benzene, which was statistically significant. Also, our positive control here, nitrogen dioxide, was decreased in viability, as was expected. We did the same for the LDH assay. We have no picture on it, but we could confirm the increase in cytotoxicity, which was concentration related. And besides cell viability and cytotoxicity, we also checked some pro-inflammatory response at the gene and the protein level. The choice of the markers was based on some literature search of ethyl benzene in vitro experiments. Here you can see the IL-8 gene expression. It's plotted and compared to clean air exposure. So on the y-axis is the log2 fold change compared to clean air. And then on the x-axis, the conditions again. It was for 5 independent biological experiments. And again, you can see a concentration-dependent increase of IL-8, which was significant for all the concentrations. And we could observe the same trend for CCL2 and IL-6 expression. We also checked the IL-8 release with ELISA. Again, we saw an increased release, which was concentration related and also significant for all concentrations. Besides inflammation, we also checked superoxide dismutase gene, but this one, it was statistically significant increase for all generated concentrations, but you cannot see a dose response here. And then heme oxygenase 1 was also checked, but it was not a relevant marker for ethyl benzene exposure. So these were some plots of the data we obtained with ethyl benzene. So our ultimate goal was to use the air-liquid interface exposed in vitro model to mimic airway exposure of petrochemical substance. To progress toward the goal, we started with a single substance, ethyl benzene. We succeeded to have an optimized experimental conditions and also to achieve a deposition efficiency that resulted in dose-related biological changes. We also did the in vitro to in vivo extrapolations, and the data demonstrated consistency in effect levels when we compared the cell viability in these air-liquid interface experiments with known in vivo effects. That was a lot of data, so we have written it down in a paper, as was mentioned. So this is now ready for submission to a journal. This was not the only experiment. So the feasibility of this method was checked also for the complex mixture gasoline. Yeah, for today it was no time to also present these data, so this will be for another time. But we prepared posters, so you can find on the SOT website, which was shown in the beginning by Amy. We have an e-poster on the gasoline data, and it's also written down in a paper and will be submitted soon. And this year, the focus will be on naphthalene, but more a mechanistic AOP-related approach will be, the goal. So these were our 2 case studies. This work was sponsored by PETA and ExxonMobil. We really want to thank them for their expertise, their critical reviews on our reports and our papers we are writing, and also the really fruitful discussions. It helps us really to move forward in setting up these systems and to try to find the methods for inhalation toxicity testing. At Vitro, we are working in a multidisciplinary team to achieve these goals. So on the picture, you can see the core team. Within the middle, my colleague Evelyn, who is also online and can help on some aerosol questions if there are some, and myself. And on the right and the left, you can see our handy, very creative technician, Anne-Ayo. And of course, we get support from other colleagues of Vitro. We have some technicians performing analytical work, backup technicians for gas exposure, molecular techniques, statisticians. We have some colleagues who have expertise in vitro to in vivo extrapolation, AOP development. And last but not least, we also have Sven Verkouteren as a business developer, and he helps us also to make a bit of shift to inhalation toxicity testing towards the use of this platform for pharma research as well. So that was it. Thank you for listening to these data.
Great. Thank you very much, Sandra. Very comprehensive presentation. Any questions for clarification before we move on to our final presenter? I'm not seeing any chats to the everyone. Stephen, do you have any direct chats to you or any hands raised that you're seeing?
We do not have any direct chats, and I am not seeing any hands raised. So let me give it one more last scan. Clive has a question.
Looks like Clive is asking a question. So Clive Roper.
Yeah, I've unmuted Clive.
Can you hear me there?
Yeah.
It was just one small thing. At the very beginning, it was suggested that this might be regulatory acceptable. I would love that to be the case, but I suspect that's all down to a regulator.
Yes, of course. This is only the beginning, of course, and with help of all of you, we can fine tune these methods and these essays, and yeah, we hope we can find the same results when testing more than one compound, of course, and to this must be tested for a full set of compounds and also tested in different labs with different systems. So we are not there yet, I think.
Even aside from that, it still doesn't make it regulatory acceptable. We can validate till our brains fall out, but actually it doesn't make it regulatory acceptable. That was just my one comment. I'd love it to be. I'd love you to be correct, but I think we need to be continuing to demonstrate to the regulators that these are, and of course that they're becoming more and more open to these norms.
Yeah.
I think it's a bit difficult to say it's regulatory acceptable when we find so many times doesn't matter how great we work on, don't fulfill the requirements of that data, of that authority. So, yeah, but I'd love you to be correct.
Okay.
Yeah, great comments, Clive, for sure. And hopefully, as we go through the discussion questions, we'll lead toward at least movement towards that path. Okay, seeing no other clarifying questions, Amy, you want to introduce our third and final speaker, Shannon?
Sure. Thanks, Dave, and thank you, Sandra, for the presentation. The next speaker is Shannon Krieger, who is a toxicologist at the Dow Chemical Company. She joined Dow in 1995 and has held various roles in Dow's toxicology and environmental research and consulting organization, the majority of those in the respiratory toxicology group, where she gained expertise in inhalation toxicology, exposure science, dosimetry, and toxicological modes of action. Her current research efforts include development of in silico and in vitro models to support new approach methodologies for respiratory toxicology, irritation, and sensitization.
Thanks, Amy. And you can hear me okay?
Yes.
Perfect. So I'm working on sharing. It's taking its sweet time, of course. Just one second. So while that's thinking about whether it's going to cooperate or not, just wanted to say hi, everyone. So very happy to have a chance to share with you all our approach for integrating new approach methodologies to assess the risk to human health for inhaled materials. Our approach is really a combination of efforts from two groups within Dow's Toxicology and Environmental Research and Consulting Organization. So myself and Dr. John Hodgkiss, who are part of the Respiratory Toxicology Group. Actually, slight correction, as of the end of March, John is officially retired from Dow, so congrats to John. And then also our Cheminformatics Group, which is led by Dan Wilson. It's still taking its time, Amy. So I'm going to just try separate. Oh, there it goes. Got it. All right. So we're combining our efforts with the two groups to to develop an integrated approach to assess new materials using in silico approaches to identify mechanism of action, and then in vitro methods to assess exposure responses. Okay, so what is the goal, right? So ultimately it is to assess the risk of inhaled materials to humans, and traditionally, historically, the in vivo rodent model has served as a test system to assess the effects in humans. But we know that the rodent model has its limitations from a relevancy standpoint, as there are dissimilar from humans in many important ways. And then also from an animal use standpoint, right, the goal really is to make use of in silico and in vitro alternative methods to directly assess the impact on human health. So inhalation testing, so why is it important? Well, we know that inhalation is a major route of human exposure, and it represents a unique interface between the environment and the systemic circulation, with uptake and potential responses in both the upper and lower respiratory tract. Inhalation testing allows us to generate exposure response data for hazard identification, with the ability to understand the integration of the properties of the material and its deposition, transport, and ADME. And then with the inhalation route of exposure, responses can be twofold, right? So portal of entry effects, where cells of the upper and lower respiratory tract are affected, and then systemic effects on other organs and tissue. So I'd be remiss if I didn't touch on dose a bit. For toxicology studies, dose is key, right? But for inhalation studies, dose can take on a little different meaning. And what we usually talk about is inhaled dose, which is the concentration of the test atmosphere times the respiration rate times the duration of the exposure. And this can really differ depending on the species being tested. And we always assume with inhaled dose, 100% of the inhaled material is deposited and absorbed. And so it's a very conservative approach. But we can refine that estimate of dose by looking at deposited dose and taking into account the actual fraction deposited. So this is better and often quite good for aerosols where we can use in silico predictive models like MPPVD. To even further refine dose estimates, we can look at absorbed dose. And this is really the mass transport of the test material into and across the epithelium times the deposited dose. And so this is an even better estimate of dose, but it requires knowledge of regional deposition and mass transport. And the dose can really be modified by local metabolism and those specific cell populations. But it's really dose to the cell that we're interested in, so cellular dose or dose per unit area. And it's here that in vitro systems offer an advantage, because as we move into these in vitro systems, we can get an understanding of the dose to the cell. Let me just see if I can move this out of the way. Yes. All right, so in our laboratory, we focused on using human 3D airway models. And we currently use both large and small airway models from Epithelial. So these are their mesolayer and small air models. Of course, vendors such as MatTech also supply these 3D alley cultures. And what they are fully differentiated airway epithelium with the airway cells growing at an air-liquid interface on a microporous membrane.
Ja, gut. Könnt ihr mich hören?
I'm sorry? Okay, so the systems we used for this work also included fibroblasts in the basolateral compartment. So these are grown on the basal side of the membrane. So we actually use the mesolayer HF, HF is for human fibroblasts, and the smaller HF models from Epithelial. So the epithelial cells are at the air-liquid interface, as they would normally be, right, in an in vivo situation with beating cilia and active mucus secretion. And they are fed from the basolateral surface, so that's depicted here. You can see that liquid compartment, and that provides really essentially two compartments to measure exposure-related effects, right? We've got the apical side of the cell culture as well as the basal compartment. So during exposure, we assess the effects of the test material on the permeability of the airways by measuring TEER, so that's trans-epithelial electrical resistance across the membrane. We also look at the viability of the cells by measuring metabolism of rhizazdrin to rhizarufin. We can also assess morphology of the cells and changes in ciliary beat frequency. And while we didn't do it for the experiments that I'll show results from today, it can also be useful to look at particle clearance or gene expression changes. And so the models allow that measurement of those endpoints as well. And then off on the right there, the basal compartment, we can also measure, as I said, other endpoints. So we measure for cytotoxicity using the LDH assay, and also expression of inflammatory cytokines. And then the basal compartment can also be used for analysis of parent test material or any metabolites. There. So regarding exposure methods, we essentially have three methods we can use. So the first is direct application of material to the apical surface. This is done with a pipette using a small volume. The second is that the test material can be delivered as a liquid aerosol. So we utilize the cloud exposure system from Vitrocell, which generates an aerosol that then settles directly on the cell cultures. And the mass that's deposited is that can actually be measured and monitored using a quartz microbalance. And then lastly, for dry powders, gases, or vapors, we can use a continuous flow system, which is really a modification of the cloud system, and it's also available from Vitrocell. So in order to characterize the responses of the epithelial cell models in our laboratory, we designed an experiment to assess the effects of a direct-acting toxicant, SDS, on the epithelial cultures. So as I said, we utilized the larger airway model as well as the smaller airway model, so mucillary and smaller from epithelic. The smaller model actually includes metabolically active club cells. And then we used the models that included the human fibroblast. So in terms of study design, the inserts were assessed prior to exposure using a measurement of TEER as well as a morphologic assessment to determine their suitability for study. And then on day 0, the test material was applied, so the test material of SDS was applied either by direct pipette or cloud exposure. And then 24 hours later, on day 1, the inserts were washed and TEER was measured as well as cytotoxicity. So again, this is LDH assay, cell viability using the rhizazdrin assay, and then we measured inflammatory cytokines IL-6 and 8. So the cultures were then allowed to recover for 1 week. On day 7, we then measured the same endpoints, TEER, cytotoxicity, viability, IL-6 and 8. Then the cultures were then fixed for histopathologic evaluation. So as I said, we either use direct application by a pipette or aerosol exposure using the cloud system to assess the differences in the responses based on application route. So essentially looking at dose rate of the test material. And the doses were chosen based on and refined based on range finding studies to find the range that gave us a good dose response. So the doses listed in the table for the 2 routes, the first 2 columns are millimolar concentration, but we've also presented them as dose per unit area or micrograms per square centimeter, which would enable us to compare the responses across the 2 dose routes. So our N was 3 per tissue type. We always include an incubator control as well as the vehicle control. Okay, so in terms of results, this slide shows the results from our range finding study with SDS. So it's a very busy slide, so I'll walk you through it a bit. The top row depicts data from mucillary, so these are the large conducting airways. The bottom row are the small air culture data. Again, those contain the metabolically active cells. The first column is TEER, for both tissue types. The second, the cytotoxicity. Again, this is LDH assay. Third column is viability. The blue bars represent day 1 data. The green bars represent day 7, or our recovery data. So if we're looking at the top row for mucilaire, so the first panel, first column is here, and it's measured in ohms square centimeter, and the line that's drawn there is at 100, which represents a significant biological effect. So below this point, you have a pretty severe effect. You can see changes above this point, but if you drop below this point, it really indicates a loss of integrity of the epithelium. The second panel, second column, cytotoxicity. So here this is presented relative to control, and the higher the bar, right, the more cytotoxicity, so 100% is about right there in the middle. And then finally, in the last column, again, these data are presented as relative to control. The viability is different here because where we have 100% viability, so as the bars go down, right, you have less viable cells, and so a decrease in that ability to metabolize the resasurant. So that's how that's measured. Along the bottom axes are the doses delivered to the cells, so dose per unit area, again, is how we presented the data. The P indicates pipette applied doses, and C indicates cloud applications. And then finally, the 2 boxes drawn in orange and red represent similar concentrations between the 2 different dose routes. So it kind of just helps guide your eye to where we saw some patterns. So if we go now and look at the data, the tier responses, if you look at the large airway responses, at the lower doses, so that grouping, that first grouping, we get an effect at day 1 with recovery by day 7. So again, day 7 are the green bars. So we had injury and we saw recovery. And at the higher doses, we saw a significant drop in tier, but no recovery. So if we only looked at the tier values in those data, we wouldn't know the underlying basis for that change. And indeed, if we look at the cytotoxicity and viability data for those cells, we do definitely get more information. So the cytotoxicity data tell us that at lower concentrations, we saw an initial release. So again, day 1, blue of LDH, but 7 days later, we were back to basal levels. But if we look at the cell viability data, we can see that for these lower doses, it makes sense because we see small changes in viability, but recovery to near background levels. If we look at those higher concentrations, we see significant cytotoxicity response. So again, in this middle panel. But the reason it doesn't come back is because, not because they're not healthy, but rather because they're almost all dead at this phase. So if we didn't have, of course, the cytotoxicity and viability data, we wouldn't be able to interpret as well those data from the tier. So we always combine all of these endpoints to really understand what the data are telling us. And then for the small air, just generally, again, we got a dose response. So this is the bottom row of data. Dose response in terms of tier, cytotoxicity, and viability. But compared to the large conducting airways, the small airways are actually a little more robust in terms of their ability to handle injury resulting from SDS exposure in our experiment. So these changes are associated with time-dependent release of cytokines. So we measured IL-6 and 8. So that's what's depicted on these graphs here. Same thing, right? Day 1 is in blue, day 7 is in green. IL-6 is in the first column, IL-8 in the second. Top row is mucilaire, so the large airways, and the small airways are in the bottom row. So here we do have vehicle control included, so that you can see baseline expression levels. And we do see a concentration-dependent difference in expression of IL-6 and 8. But again, the small airways are seem to be more robust and have a greater and more sustained release of IL-6 and 8. So we did find that these physiological responses had morphologic correlates. So these are light micrographs of large airway on the top row and small airway on the bottom row. So the left side of the figure are cells stained with Alcium blue and H&E. So Alcium blue will stain the stored mucous substances. And so if we're looking at the left side, that first column is control, and the second column are dose cells. For the control, you can see a nice thick epithelium. You can see the microporous membrane on the slide, and then the fibroblasts underneath. So we can see that in the dose cells, we see a reduction in the overall epithelial thickness compared to control, as well as the stored mucous substances. So again, these are these kind of blue blobs, right? It's the stored mucous substances. So we had a release of stored mucous substance, as well as a loss of cilia. And then on the right side, so we can also look at whether there are changes in cell proliferation. We use Ki-67 as an indication of cells undergoing division. And so the right side of the figure shows this. For the large airways, there doesn't appear to be much of a change in the number of cells undergoing proliferation. But for the small airways, there appears to be an increase in the labeling index. And so these are qualitative results at this phase. For the range finding experiment, we did not do a quantification of the Ki-67 labeled cells. But the results, right, show that although the cells underwent an injurious response to SDS, they were capable of regeneration. So to confirm and expand the results of that first study, when we undertook a second study with the same design, but this time we actually matched the exactly the dose per unit area between the two routes. So the definitive study described down here, if you look at this dose per unit area, right, these are matched between the two dose routes, the cloud and the pipette. And you're already familiar with the layout, so I won't walk you back through, but what we saw were reproducible results. Again, figures laid out the same way. Mucilars on top, smaller on the bottom. But I don't have the boxes here, but you can see that there's similar pattern of responses at the same dose per unit area, regardless of dose route. So overall, so again, right, cloud and pipette for 25, 50, and 100 are depicted paired together. And the same is true then of our results for the inflammatory cytokine release. So for this study, we did quantitate the Ki-67 labeled cells to assess cell proliferation. So here we have mucilare on the left, small layer on the right. There's two routes, right? The cloud is in blue, the pipette's in purple. And it's here again that we see the same as when we saw on the micrograph that I just showed in the previous study, that the small airways are undergoing more of a proliferative response than the larger airway cultures. So overall, the conclusions from our studies indicate that we see a similar exposure response profiles across the two dose routes. So this is when the dose per unit area is equivalent between the two routes. The smaller appear to be more resistant than the mucilare to the direct acting toxicant that we applied, SDS. We feel that our results underscore the need to measure multiple endpoints to kind of better characterize exposure response profiles. And then, unlike traditional in vivo studies, deposited and absorbed dose and non-exposure concentration is what's important when we're looking at in vitro test systems. So Amy, I can stop here. I've got a couple more slides just touching on in silico, but if you'd like, I can end things here. Shall I keep going?
Okay.
Amy, Dave?
Yep, sorry, thanks, Shannon. Dave, what do you think, looking at this time and the questions that you've had come in?
So we don't have any questions so far.
Well, then maybe I should stop. It's up to you guys. I don't have to touch on the in silico piece. I plan to, but I know that we're a bit over already, so.
I think if it's just a few minutes, I would go ahead, Shannon.
Okay, sure, I'll breeze through it. Okay, so now that I've just described our in vitro system we're using and some of the studies we've conducted, I just wanted to briefly touch on the incorporation of in silico approaches in our integrated approach for assessment of inhaled materials. So for the past several years, our cheminformatics group has been curating data and building in-house profilers for various mechanisms. There's some listed here on the left. It's actually not all-inclusive. There's been several that have been added recently, including those for sensory irritation. And then they've also been building docking-based models, and those are listed on the right, again, not inclusive. But because we have these valuable tools, we've proposed an inhalation IATA approach, which involves first and foremost, so at the top, in silico identification of those molecular initiating events, so we can use our in silico tools to do that, and then alignment of that MIE and AOP based on our predicted and observed outcomes. And then that will really drive selection of our in vitro model based on those predicted key events. And then the thought is that we will proceed right with evaluation of in vitro exposure response and evaluate that based on a predicted toxicity in silico. And that's kind of an iterative process. So really what that looks like is we'd be doing computational mechanistic profiling to determine an MIE initially, and then determine whether the MIE is preferentially specific to the inhalation route versus other routes of exposure, such as oral. So if it is, obviously we would make use of our specialized in vitro models. If not, then something that's more route agnostic or potentially assess bioavailability if it were oral route. Ultimately, the goal would be to simplify and standardize these systems, right? Because we know that that would be key to regulatory acceptance. And then just lastly, again, I'll hit on that knowing that tissue dose, right, in vitro and in vivo is really will ultimately determine if that exposure response is equivalent across platforms. So just thinking about exposure concentration when moving into these in vitro models is not sufficient. We feel that to determine their value for human health risk, we really need to look at the tissue dose. So now I'm done. Thank you.
Great. Thanks a lot, Shannon. That was very helpful. We did have one question that came in just before you started on the in silico related to measurements of transepithelial electrical resistance. Is it possible to determine whether changes in recovery are due to the changes to the epithelial cell layer or the fibroblast, since they both appear to change?
Yeah, and I think histopathologically, right, we have the opportunity to do so, but the tier measurement itself will not, right, inform you as to which of the cell types are doing that recovery or which of them are hit. That's really more histopathologic evaluation would be required.
Okay, great. I'm not seeing any additional questions. Stephen, any hands raised?
Yes, I've got one from Holger, and I've just unmuted them.
Okay.
Hi, yeah, this is Holger from IIVS. So just a quick question about the mass per area deposition conversion from the pipette dose. So obviously, you deliver a certain volume of material solubilized in a buffer, and then you had that one table where that was translated into mass per square centimeter. Is that something, or is it considered 100% bioavailable? That is everything that was dissolved in that material. And then the second question I have is, was mucil air and small air from the same donor?
Yes, it is considered 100% dissolved, Holger. So we make that assumption when we're doing the calculation. So very good question, thank you. And then yes, these were from the same donor for both studies.
Thank you.
Okay, great. And one quickie before we break. Shannon, have you published the in silico work on the MIEs? And is it comparable to the method developed and the approach done by Cambridge University or Tim Allen on 39 targets? They've now expanded to about 80.
So we've published some of it in pieces, right? It depends upon the profilers and the models. So some of those have been published, some of them are currently, those manuscripts are being worked on. And I don't think I'm going to be able to answer how it compares to the other work because I'm not familiar with it.
Next up, we have brief comments, just about 5 to 10 minutes each, from 3 U.S. government agencies. First up, the DoD, and then 2 from EPA. And as we all know, and Clive pointed out earlier, developing approaches that meet the needs of industry and government and regulatory agencies is very important. So these perspectives will help put what we've talked about so far into a regulatory context, at least from the perspective of one regulatory agency. So we will start off with Dr. Emily Renke, who is a biologist with the Health Effects Division of the U.S. Army Public Health Center and co-chair of the Interagency Coordinating Committee on the Validation of Alternative Methods, or ICVAM. Emily?
All right, can you guys hear me?
Yes.
Okay, perfect. Can you see my screen? Because can't quite tell. You can see it. Perfect. Yay, everything is working. So good morning, everyone, and thank you, Amy and Dave, for putting this together. I'm just going to give a brief overview from more of a non-regulatory but federal standpoint of what DoD needs for inhalation toxicity testing. So quick glossary, just in case anybody doesn't know what all these mean, but I'm sure you do. And a quick overview of the needs that DoD as a whole has. We collect a large variety of data from LC50s all the way up to reference concentrations, occupational exposure levels, and then we extrapolate from our animal data to human exposures. We have acute through chronic endpoints, including reproductive and behavioral that we're interested in. Our chemical classes are wide. We have organophosphates, high nitrogen compounds, including that would be like a pyrotechnics and explosives. We have to worry about fuels, we have to worry about propellants, fire extinguishing agents, and combustion products. And our exposure scenarios are gases, vapors, aerosols, which include the droplets and mist, and particulate dust. And we might be in some non-traditional scenarios, such as high altitude, high G-force, high pressure, a recirculated environment. You might be under hypoxic conditions or have to worry about extreme temperature and humidity, as well as the ever-present mixtures. And our hazard categories are low toxicity through to high toxicity. So as you can see, DoD just pretty much needs everything, which doesn't make this easy. Now a quick breakdown of the various different services. So if you're interested in helping, Army with more of a ground-based support or Air Force with air-based support, then that's where that would be coming in. So Army, again, we have acute through chronic durations. We're concerned with acute lethality, local effects, air-blood barrier breaches, as well as systemic effects. And that acute lethality is going to be that, really lethal exposures. Information needs, we need LC50s, LCT50s, IDLHs, OELs. We need to make those PPE determinations. We're trying to make some slope calculations, our toxic load exponents, and then again, just kind of BMDLs, OELs, OELs. For the more traditional toxicity assessments. Again, those chemical classes, very similar to what the whole overview is. And we also have to worry about metabolic poisons specifically for Army as well. And again, those same exposure types, gases, vapors, aerosols, and hazard categories, again, the whole gamut, non-toxic through highly toxic. Air Force, again, same duration, acute to chronic. Acute, they're looking for AEGL and dose determination for longer term studies. assay endpoints, local effects, irritation and inflammation, and systemic, inclusive of behavioral effects. Again, if you're worried about somebody up in the up in a jet and they're having some inhalation issues, you don't exactly want them losing ability to fly a plane. Information needs, we need OELs, AGLs, and again, those PPE determinations. Chemical classes specific to Air Force, fuels and propellants, liquid propellants as opposed to powder propellants, engine oils, coolants, hydraulic fluids, and heavy metals. They also have big concerns with the fire extinguishing agents too, which I forgot to put on here. Exposure types, again, gases, vapors, aerosols, and that low toxicity through highly toxic hazard category. Navy, same story, acute to chronic. They're more interested in local effects such as irritation, inflammation like Air Force, and systemic effects including physiological, behavioral, and cognitive, and then reproductive and developmental effects. Looking for OELs, AGLs, CELs, EELs, and PPE determinations. And their more specific chemical classes are fuels and propellants, coolants, heavy metals, solvents, combustion products, and emissions. And again, gases, vapors, and aerosols, and low toxicity through to high toxic. So what are our commonalities? Everybody needs everything from acute to chronic. We're looking both at local and systemic effects, including reproductive and developmental and behavioral and physiological changes. Really, the big thing is PPE determinations and OELs, and then some acute hazard determinations. The chemical classes are a wide range. We have the industrial chemicals, chemical agents, heavy metals, energetics, fuels, the full exposure gamut, the full hazard category gamut, and what we currently have, just to kind of sum up what we're currently doing, is we have non-animal approaches through QSAR modeling, some toxicokinetic models, and some cell-based and tissue chip models is kind of where we stand right now. However, we're consistently trying to expand our approaches as we go forward. So with that, pretty slide that they make me put on there just so you can see everything we do. And there's my contact information if you need any additional information. And with that, that's all I have.
Thanks, Emily. We will jump right to the next presenter now, which is Todd Stedford, who is the Senior Science Advisor for EPA's Office of Pollution Prevention and Toxics.
Okay, so I'm going to talk about new approach methodologies and the context of use under the amended Toxic Substances Control Act, or Tosca. And slide to, there we go. Okay, so Tosca in 2016, Tosca was amended, and there's two primary categories of chemicals that are evaluated as far as industrial chemicals go. They're designated as new chemical substances or existing chemical substances. And the difference between those two is that existing chemical substances are on the Tosca inventory. They can be manufactured, introduced into the stream of commerce if they're not already there.
Slides.
Yes. Or they were, and they've actually undergone the new chemical review process, or they were grandfathered in when Tosca was passed in 1976. And some of the amendments for new chemical substances on the left side of the screen, EPA now has to make one of 5 risk determinations. And the first is whether the chemical or a significant new use of the chemical presents an unreasonable risk of injury to health or the environment. The second is that the available information is insufficient to allow the agency to make a reasoned evaluation of health and environmental effects associated with the chemical or the significant new use of the chemical. The third is that in the absence of sufficient information, the chemical or significant new use may present an unreasonable risk of injury to health or the environment. The fourth is what's known as an exposure-based determination. It's where the chemical is or will be produced in substantial quantities and either enters or may enter the environment in substantial quantities, or there is or may be significant or substantial exposures to humans. And the fifth is the chemical or significant new use is not likely to present an unreasonable risk of injury to health or the environment. Now, on the existing chemicals space on the right side, EPA is now required, per the amendments in 2016, to prioritize existing chemical substances and to make designations as to whether they're considered low priority or high priority substances. And if EPA determines that a chemical substance is a high priority substance, it has to perform a risk evaluation and make a determination as to whether it presents an unreasonable risk of injury to health or the environment. Now, as far as review of existing chemical substances at EPA, new and existing chemical substances, EPA must make risk-based determinations, evaluations that are based on the best available science, the weight of the scientific evidence, and without consideration of costs or other non-risk factors. And in doing so, EPA assesses a wide variety of potential human health and environmental effects associated with the known, intended, and reasonably foreseeable conditions of use for both new and existing chemical substances. So the types of data that are utilized for these reviews: for new chemical substances, submitters must provide all data in their possession or control related to health and environmental effects, and EPA may require the development of new information relating to the new chemical substance. So that would relate back to determination that there's insufficient information. For existing chemical substances, EPA must first take into account reasonably available existing information, and EPA may also require the development of new information relating to the existing chemical substance. And as you can see on both of these paths, for new or existing chemical substances, there's an opportunity to review existing data that was generated using new approach methodologies, or to require the development of data utilizing new approach methodologies. So one of the additional amendments under Tosca that really drove the use of new approach methodologies, Section 4, and the reduction of testing on vertebrates. And under Section 4, the administrator of the EPA is required to reduce and replace, to the extent practicable and scientifically justified, the use of vertebrate animals in the testing of chemical substances or mixtures. And there's also a provision that addresses voluntary testing and states that if the administrator of the EPA has identified such a test method or strategy for the development of such information, that the entity conducting the tests and generating the data would use that alternative test or new approach methodology before conducting new vertebrate animal testing. So the use of new approach methodologies under the amended Tosca, OPPT, which is the office within EPA that has the delegated authority with administering Tosca, they've utilized various new approach methodologies to exclude chemical substances from specific chemical categories. Chemical categories are those that have identified toxicological concerns for particular chemicals. One example is with polymer lung overload. And recently, on April 1st, 2020, EPA issued a proposed rule to revoke a significant new use rule, or Sner, for new chemical substance. And that revocation was based on the results of a biosolubility study. And this is significant because it represents the first time that EPA has utilized a new approach methodology for revoking a Sner, or proposing the revocation of a Sner. And the original Sner would have required a subchronic inhalation toxicity study in rats in order to use the chemical substance in a manner that was inconsistent with the original new chemical substance submission. Now, some of the general questions that we receive are: what approaches will OPPT consider in support of making a risk determination on a new chemical substance? And OPPT, they do maintain a list of alternative test methods to animal testing, which is available at the URL shown. Another common question is, will OPPT accept data for a method that is not on the list of alternative test methods? The answer to that's yes. EPA considers any and all data that's provided in a submission. However, EPA does review the data for reliability, quality, relevance, and EPA also encourages submitters to consult with EPA before conducting any testing, and they have a general process for doing so. It's called a pre-submission meeting, which the details on that are provided at the URL shown. Thank you.
Thank you, Todd. And our last prepared comments for today, before we get into the Q&A and discussion, are from Dr. Monique Perron, who is a senior toxicologist in the Health Effects Division of the EPA's Office of Pesticide Programs.
Good morning. I'm just waiting to share my screen. Okay. Hopefully my screen is showing now.
It's not
It
yet
looks like
on
it's
my
in progress.
end.
Yeah, looks like it's starting to.
Okay.
Did you hit share with whatever it is that you're trying to share?
Good question.
It's just details.
Oh yeah, sorry, I didn't do that part. That would be it.
I know that's from experience.
Yeah, always seems to happen, right? All right, so hopefully now you can see my slides.
Yeah, we can see them great. Thanks, Mike.
Okay, wonderful. So yeah, I'm just going to give a quick overview of what we look for inhalation studies in the Office of Pesticide Programs and some of the efforts that have been made to try to refine or replace those studies. So under our the Code of Federal Regulations, Part 158, we require acute inhalation toxicity studies, not only on the technical active ingredient, but also on formulations or end-use products. Additionally, we were required a 90-day inhalation toxicity study on the technical active ingredient. If you go to the CFR, it says that this is conditionally required. However, there's a footnote that says that it's required if there's a likelihood of significant repeated inhalation exposure to the pesticide. So in most cases, for our conventional pesticides, there's a trigger from that footnote to require the study. Additionally, there's a footnote in that table as well that indicates that shorter duration studies of either 21 or 28 days may also be sufficient. But we do have flexibility in our Part 158 requirements. We have given quite a few waivers for inhalation studies using a weight of evidence approach where we consider not only the toxicity of the chemical, but also the exposure and the risk estimates to determine if we think that an oral point of departure will be protective in the cases where we don't have route-specific data. Additionally, we can consider alternative approaches that can be accepted or other data can be used in conjunction with available information to meet the requirement of that study, of the studies. And as many of you may have heard, recently in September 2019, the administrator published a memo prioritizing efforts to reduce animal testing with a goal of reducing requests for and funding of mammal studies by 30% by 2025, with an overall aggressive timeline of eliminating mammal study requests and funding by 2035. This memo also asked us to, in OCSPP and ORD, to form a working group of experts in the field who will provide a work plan within 6 months, and also to hold a workshop, which we held in December. Our first annual conference was held in December to look at the state of the science. So there's really a push for us to be moving away from whole animal studies and considering these alternative approaches and new approach methodologies when we're considering our data requirements and making a safety finding for pesticide registrations. And where we've really seen most of our growth is in the repeat dosing studies for contact irritants. So as probably many of you on the phone or on this webinar know, we brought a proposal from Syngenta that they provided for chlorthalonil, that is a contact irritant, to our scientific advisory panel in 2018. And this is a collaboration with our friends at NICEDM, as well as across the agency, having people from OPPT and ORD involved in reviewing it. And we convened our SAP partly because this would have been one of the first times the point of departure for risk using would be derived using an in vitro data. Although I think we might get beat by a different, there might be one that goes out before this one. And then also there's the potential to use this for other contact irritants. And we also were curious about the potential to use it for other chemicals that have the potential to cause portal of entry effects in the respiratory tract. So in April of 2019, the SAP report was put out, and none of the panelists said, you should stop what you're doing and go back to that laboratory whole animal study. They were very receptive and very supportive of moving forward. There were just some general recommendations that we've been working with, along with Syngenta over the past year. And the plan is to take all of the available information that we have, and currently chlorthalonil is scheduled to go through registration review in our last quarter. So we should hopefully have our first draft risk assessment for chlorthalonil out later this year. So that's in definitely for the contact irritants area, where we know a little bit about the chemical. We know that it's direct acting, it hits the cells, it causes the damage. We've also had people come to us and say, well, we have rat studies that have shown irritation effects in the respiratory tract. So what if we refine our assessment using that knowledge, but then do repeated exposures with those with the 3D model? So we have a few people who have started to look into that potential. So not necessarily a direct contact irritant, but a lot of the time we are seeing that for the chemicals that we use the inhalation endpoint, we're seeing, excuse me, the effects are in the upper respiratory tract most of the time. So this is a slide that actually we presented a little bit of this information at last year's ancillary meeting, where we just did an informal analysis of our database to see, how many chemicals have we used the inhalation toxicity study to evaluate inhalation exposures for human health risk assessment. And at that time, it was 127. And then when we broke it down, about 50% of those chemicals were portal of entry effects. Most of the time, usually it's upper respiratory tract, the larynx, the nasal turbinates. And then in the case of when we did have chemicals that had systemic effects, the majority of those were chemicals where we had a known neurotoxic mode of action, so OPs, pyrethroids, carbamates. So if you kind of look at it this way, for chemicals that we don't really know much about the mode of action, like we have a set mode of action, we're really looking at portal of entry effects that are driving our risk assessment there. And they're not all direct contact irritants or corrosive chemicals. They truly are chemicals that sometimes you have no indication in the oral studies that they would cause this type of irritation in the respiratory tract. It's a very route-specific effect that we're looking for. So from our perspective, it would be great to have some sort of tiering or framework to try to figure out, if you have a new active ingredient coming in, could you bypass needing to have a whole animal study by moving to the 3D, and what would be those, how would you determine which chemicals those would be? So trying to figure out some sort of way to maybe at the very least, reduce the number of chemicals that need to go the whole animal route before maybe hopefully the replacement of that whole animal study altogether. And I don't have any slides here on the acutes, but we've had several efforts in the past couple of years that have tried to correlate things like cell viability and tier, and a lot of these effects that we're looking at in the 3D model to our acute tox categories, and there really has been a lot of challenges with that. So I think that's where we really haven't made many strides. There's been a lot of ideas and trying to make those correlations work, and in some cases they work really well, but then it kind of sort of falls apart when you start to get away from acute tox category 1. So I think again, from a regulatory standpoint, if there are different assays that we should be looking at or different endpoints that should start becoming the focus of those types of comparisons, I think that would also be helpful for this discussion that we're having today. And I think from there, I will go back to Amy and Dave.
Great. Thanks very much, Monique, and Todd and Emily as well. It's very tremendously helpful, obviously, to have that context, those contexts of use in mind.